DNA replication starts at specific sites called origins of replication. In eukaryotes, there are multiple origins of replication, which are short stretches of DNA with a specific nucleotide sequence. Proteins that initiate DNA replication recognize this sequence, attach to the DNA, and unwind the two strands, creating the replication bubble. Replication proceeds in both directions until the entire strand is copied. At each end of the replication bubble, there is a replication fork, a Y-shaped region where the parental DNA strands are unwound. Unwinding of the parental DNA involves several proteins. An enzyme called helicase untwists the double helix at the replication fork, and the single strand binding protein stabilizes the unwound strands by preventing the formation of hydrogen bonds between the nitrogenous bases of the two strands. This untwisting of the double helix causes tighter twisting on the region ahead of the replication fork. Therefore, topoisomerase helps relieve this strain by breaking, swiveling, and rejoining the parental DNA. Once the strands are unwound, the parental strands are available to be used as template strands to synthesize new strands. Enzyme like DNA polymerase synthesizes new DNA, however, it can only add nucleotides to the pre-existing chain. Therefore, a short RNA chain called a primer must be formed first. An enzyme that joins a RNA primer is called primase. DNA polymerases are enzymes that catalyze DNA synthesis, and as mentioned before, they can only add nucleotides to the pre-existing chains. There are many different types of DNA polymerases, but two that play major roles in replication are DNA polymerase 1 and 3. Most DNA polymerases require a primer and a DNA template strand to add nucleotides to the growing strand, and DNA polymerase 3 is one of these polymerases. Nucleotides that are added to the new DNA strand comes from nucleoside triphosphate. Nucleoside triphosphate consists of a sugar and a base with 3 phosphate groups. A similar molecule that we have already learned about is ATP. ATP, like DATP, a nucleoside triphosphate that supplies adenine nucleotide to DNA, has 3 phosphate groups, a sugar and a nitrogenous base, adenine. However, while the sugar component in ATP is ribose, the sugar component in DATP is deoxyribose. DATP is chemically reactive because of the unstable group of negative charges. So as each nucleotide joins the growing strand, 2 phosphate groups are given off as pyrophosphate. This exergonic reaction is what helps drive the polymerization action. As we have learned previously, DNA strands are antiparallel to each other. One strand runs from 3' to 5' while the other strand runs from 3' to 5'. So when new strands are being synthesized, they must also be antiparallel. This antiparallel structure of DNA requires DNA polymerases to add nucleotides only to the free 3' of the primer and not the 5'. Along one template strand, DNA polymerase 3 synthesizes the complementary strand by continuously adding nucleotides in the 5 to 3' directionality. A strand synthesized in this mechanism is called the leading strand. Because nucleotides are being added continuously, only one primer is needed. To synthesize the other strand in the 5 to 3 directionality, it must elongate away from the replication fork. This particular strand is called the lagging strand, and it adds nucleotides discontinuously, creating a series of segments called Okazaki segments. Because the lagging strand grows continuously, multiple primers are needed. Now let's take a closer look at how the lagging strand is synthesized. First, the primase synthesizes the primer, then DNA polymerase 3 adds nucleotides to the 3 end of the primer. When DNA polymerase 3 reaches the primer of the next segment on the right, it detaches from the DNA. Now Okazaki fragment 1 is completely synthesized. Okazaki fragment 2 is synthesized in the same mechanism. Now the DNA polymerase 1 replaces the RNA of the primer with DNA. At this point, the last nucleotide of the Okazaki fragment 1, which is not attached to the first DNA nucleotide of the Okazaki fragment, whose primer was replaced, is joined by DNA ligase.